Shirley Seah's PhD oration. Shirley did her honours many years ago, to great, went back to Singapore, worked some time, finally convinced the Singapore government for a scholarship to come back here to do a PhD. We were the lucky lad to receive her. Shirley's great to have in the lab. She's sort of, she's assiduous, tackles things without any complaint whatsoever. Likes to be challenged with regards to new techniques, and she's sort of done quite a wide spectrum of techniques that we didn't even have in the lab, because we're not really a biological lab. And she sort of brought that in with her. And she's one of these people who sort of volunteers to help you. So if something needs to be taken out on a Saturday morning, or an extra pair of hands to process the facts, Shirley always is the first volunteer. So, and we, I think Shirley's one of the great pleasures about being a PhD supervisor is to see the development of someone as they go through the years of their PhD. Shirley came out certainly as a sort of fairly shy, but she's less shy now. But I think it was a great pleasure to see her develop over those years to become basically, an independent researcher, which is what you want to prepare to do. So, I'm going to tell you about her story because she's going to tell you about her story. Sure.
Well, thank you, Andrew, for that very kind introduction. So today I'm going to share with everyone some of the results we've got over the years and show you that, unlike CD4 T-cell help, co-stimulation is necessary for the generation of primary CD8 T-cell responses during influenza infection. I'm sure everyone is familiar with the pandemic flu that has taken place over the years, especially with this picture over here that is taken during the Spanish flu outbreak in 1980 that killed 50 million people. The latest flu pandemic we had was in 2009, and that caused about 8,000 deaths. In general, in Australia, there are about 3,000 deaths that is due to influenza virus infection and its complications. And as you can see here with the hospitalizations and GP consultations, it costs the Australian healthcare system $85 million per year, and 1.5 million workdays are lost when people get sick with flu. So during influenza virus infection, the virus starts to replicate in the host, as in this case here is the mouse. The viral load peaks at about day 5, and it starts to decrease as the adaptive immune system kicks in. So the viral clearance usually occurs around day 8 to day 10, and the adaptive immune system continues to climb and peak about day 10, and it will decrease to form a memory pool after the clearance of the infection. Studies have shown by Peter a long time ago that the clearance of influenza virus infection is due to cytotoxic T lymphocytes, or CTL for short, which are basically activated CD8 T cells. And mice that lack CD8 T cells are unable to clear virus infections, resulting in mortality. So how do CD8 T cells get activated to become CTLs? The canonical view of CTL activation was first derived using non-microbial antigens. What happened was that the dendritic cell will take up the antigen, process it, and presents the antigenic peptide on its MHC class II molecule to the T cell receptor on the CD4 T cell. This interaction also involves co-stimulatory molecules such as CD86 and CD28, and CD40 and CD40 ligand. These 3 Nature papers in 1998 demonstrated that the interaction between CD40 and CD40 ligand will result in the activation of dendritic cells, or what they term as licensing of dendritic cell. This activated dendritic cell then moves on to prime a CD8 T-cell to become a CTL. However, during some infections, such as those with influenza viruses, the infection itself can directly activate the dendritic cell, and this dendritic cell then moves on to prime a CD8 T-cell to become a CTL. So in this scenario, the CD4 T-cell help is no longer required. Now, how can a CD8 T-cell get activated without CD4 T-cell help? Does this interaction over here between the dendritic cell and the CD8 T-cell also requires co-stimulatory molecules such as those observed previously on for the CD4 T-cells? And if this is happening, then what is the real role of CD4 T-cell help in primary CTL responses? These questions then bring us to the aim of our project, where we want to find out how CD8 T-cell can get activated without CD4 T-cells help during influenza virus infection. And does the direct co-stimulation of CD8 T-cell play a role in this activation? As such, we decide to look at the CD28 and CD8086 co-stimulatory pathways and the CD40 and CD40 ligand co-stimulatory pathways. So during T-cell activation, CD28 would bind to CD8086. There is also another molecule called the CTLA4, which would also bind to CD8086. In fact, the CTLA4 binds at a higher affinity than CD28. Therefore, it outcompetes CD28's binding to CD8086. So our lab generated the CTLA4 Ig transgenic mouse, where in this mouse they produce a CTLA4 immunoglobulin that mimics this molecule over here. So the CTLA4 Ig would bind to CD8086 and it blocks its interaction with CD28. Therefore, these mice, CTLA4 Ig transgenic mice, they do not have CD28 co-stimulation pathway. We also have the CD40 ligand knockout mouse and the CD8086 knockout mouse. We use the CTLA4 Ig transgenic mice and the CD40 ligand knockout mice for in vivo assays. And we also use the CD40 ligand knockout mice and CD8086 knockout mice for in vitro experiments. And I'll explain to you later on why we do not use CD8086 knockout mice for the in vivo experiments. So the CD8 T-cell recognizes two dominant epitopes on the influenza virus. The first one is found on the polymerase acidic protein, or PA for short, and the other is found on the nucleoprotein, or NP for short. And in our project, we decide to focus on the PA. To detect antigen-specific CD8 T-cells, or PA-specific CD8 T-cells in this case, we used a technique called tetramer staining. The beauty of this technique is that not only can we detect CD8 T-cells during influenza virus infection, we can also detect very low numbers of antigen-specific CD8 T-cells. For example, when the mouse is at naive stage, so we can determine the naive precursors within the mouse before we start any experiments. So what are tetramers? Basically, tetramers are just 4 of these MHC class 1 molecule containing the PA epitope conjugated together by streptavidin. So that's our tetramer. And we use this tetramer to detect PA-specific CD8 T-cells. During influenza infection, the number of CD8 T-cells, or antigen-specific CD8 T-cells in this case, can increase 50-fold by day 7 post-infection and up to 3,000-fold by day 9 post-infection. So during this course of infection, the number of cells can start from 100 at naive state and increase to a few million during acute phase. So if we want to look at the immune response during infection over the course of these few days, we need to find an enrichment protocol that can accurately isolate the number of antigen-specific CD8 T-cells, both at this time point and also at this time point. So what we did was to compare two existing protocols, which is commonly used. One is the negative enrichment protocol, and the other is the positive enrichment protocol. So what is the negative enrichment protocol? What we did in this protocol is firstly to take infected cells and incubate them with a depletion cocktail that contains antibodies that are specific to the cells that you are not interested in. And we are able to pull these cells out using a magnet, as you can see here. And the cells that you want are actually free, and so we just collect these cells and continue to stain them with PA tetramers and other surface staining. What is positive enrichment in this case? Again, we take infected cells, but this time we incubate them with PA-specific tetramers. And because these tetramers are conjugated to PE, we can pull these cells out using magnetic beads that is specific for PE. And on the magnet, as you can see here, the cells that we want will now bind to the magnet. Cells that you are not interested in can be washed away. We harvest the cells that we want by removing the sample from the magnet and eluting the PA-specific CD8 T-cells, and we continue with our surface staining. To compare the two protocols, firstly, we take splenocytes from an infected mouse, prepare single-cell suspension, and because at this time point, the number of PA-specific CD8 T-cells is so high in this sample, we can directly stain for PA-specific CD8 T-cells within this sample without the need for any enrichment. Then we take equal numbers of cells, one for the negative enrichment and the other for the positive enrichment. We then dilute the sample 1:4 with naive spleen cells. And at this dilution, again, we take equal numbers of cells out for each of the enrichment protocol. We continue to do the dilutions with naive spleen cells, and at every step of the dilution, we will take out equal number of cells for either negative enrichment protocol or positive enrichment protocol. As mentioned earlier, because we are able to stain directly for the number of antigen-specific CD8 T-cells within the sample that is not processed, we know the number of starting material, we know the number of starting PA-specific CD8 T-cells within the material, which is our number here, and we take that number and divided it by our dilution factor, hence generating a theoretical value that we're supposed to have at every dilution, and thus giving us the standard curve. We next plot the results we obtained from negative enrichment against this standard curve, and we found that when the number of cells are limiting or low in numbers, especially, for example, you can see here about 1,000, this enrichment protocol actually provides or gives you a false positive result, and it predicts, gives you a higher number than there actually is within the sample. Hence, this enrichment protocol is not suitable when your target cells are low in number. We next look at positive enrichment, and we found that this protocol is actually pretty good. It can not only detect accurately cells that are high in numbers, it also can give you a pretty good results with a sample that has low numbers of target cells. So in order to do our experiments, we decided to use this enrichment protocol to look at PA-specific CD8 T-cells in a naive mouse and during infection. So what we did was basically take the wild-type black 6 mouse, or CD40 ligand knockout mouse, a CTLA4 Ig transgenic mouse, deplete them of CD4 T-cells, infect them with influenza A viruses. Now, before we look at antiviral response, we first look at the number of antigen-specific CD8 T-cells within the naive mouse, and I'll explain to you why we do that later on. And we also look at the viral load in the lungs of these mice. Again, I'll show you why we do that. And for antiviral responses, we look at the number of antigen-specific CD8 T-cells at day 8 post-infection, and we also look at viral clearance. So as promised, why do we measure the number of antigen-specific CD8 T-cells in a naive mouse? Firstly, it has been shown that CD28 is important for the intrathymic development of T cells and invariant NK T cells. So all the previous studies that are done by other researchers using knockout mice and infection model, they have never checked if the genetic modification actually affects the development of their T cells, especially in those CD8 T-cell knockout mice or in the CD28 knockout mice. So they are not sure if their final readout is accurate or is it biased towards the number of starting material that they have within the mouse. So for us, we need to ensure that all our genetically modified mice actually have similar starting number of PA-specific CD8 T cells. Luckily, they are the same. So, as you can see here, the CD40 ligand knockout mice and the CTLA4 Ig transgenic mice, both of them have similar number of PA-specific CD8 T cells within them as compared to the wild-type mice. But, as I mentioned earlier, why we don't use these mice? These naive CD80-86 knockout mice, they actually have half the number of PA-specific CD8 T cells in them as compared to these other three groups of mice. So that is why we do not use these CD80-86 knockout mice in our in vivo studies. We also want to ensure that the virus is able to replicate as efficiently in all the groups of mice. So we check the viral load at day 5 post-infection, and we find that the virus titer within the different groups of mice is actually equal. So the infection is equally robust in the different groups of mice. So that's all good. Now to the crux of the experiment. Does co-stimulation affect the generation of anti-influenza CD8 T cells? So, and when we look at day 8 post-infection, we found that mice that lack CD40 ligand co-stimulation and CD4 T cells here, as you can see, the number of PA-specific CD8 T cells is reduced. Likewise, for mice that lack CD28 co-stimulatory pathways, their number of PA-specific CD8 T cells is also reduced. The most interesting thing is, when we look at these results here, if we leave the CD4 T cell compartment intact within the different groups of mice, you will notice that their presence or absence actually did not alter the ability of these mice to generate PA-specific CD8 T cells. For example, even if CD40 ligand is missing in this CD40 ligand knockout mice, you can see that the numbers between these two groups are actually similar. Same trend for the CTLA4 Ig transgenic mice. So the result so far shows that, firstly, the direct co-stimulation is important for the generation of helpless CD8 T cells, and secondly, CD4 T cells do not play any role whatsoever in the generation of primary CTL. Now, when the CD8 T cells get activated, they upregulate surface molecules such as CD44, CD25, and CD69. So to look at the effects of co-stimulation on the activation of antigen-specific CD8 T cells, we decide to focus on CD44. And to look at proliferation of these CD8 T cells in the absence of co-stimulation, we decide to use bromodioxyuridine, or BRD for short. Now, BRDU is an analog of thymidine, and this will get incorporated into proliferating cells. So with this method, we can track the proliferation of CD8 T cells. When we look at the activation of these cells using CD44 staining, we found that when CD28 co-stimulation is being blocked, these cells have an impairment in their activation as compared to wild-type mice. However, when we look at the proliferation of these cells using BRDU staining, we found that once these cells get activated, they are able to proliferate as well as the wild-type mice. This result therefore shows that co-stimulation actually affects the activation of these CD8 T cells, but they do not affect their proliferation once they get activated. So what happens with CD40 ligand? In contrary, we found that the loss of CD40 ligand co-stimulation actually do not affect the activation of CD8 T cells, nor affect their proliferation. How about viral clearance? Right. As you can see here, mice that have their CD28 co-stimulation blocked are not able to clear virus infection as efficiently as the wild-type mice over here. And the loss of CD40 ligand co-stimulation just results in slight delay in viral clearance, just 1 or 2 mice, but not a significant delay. So again, this result demonstrates that CD28 co-stimulation is important for viral clearance. What happens if there's CD4s? Again, we look at the presence of CD4 T cells within the CTLA4 Ig transgenic mice, and we found, like the previous studies for CD8 T cells, their presence or absence actually do not alter the ability of these mice to clear virus infection. Hence, again showing that CD4 T cells actually do not play any role in viral clearance. So the summary so far for the day 8 studies. Firstly, positive enrichment is the most suitable protocol. And all groups of mice have the similar number of naive PA-specific CD8 T cells. The direct CD28 and CD40 ligand co-stimulation of antigen-specific CD8 T cells are critical for generation of primary CTL responses and also for viral clearance. And lastly, CD4 T cells do not play any role in the generation of primary CTL or viral clearance. So what we've seen so far are all day 8 post-infection. Then it brings us to the question now is to think about, does co-stimulation takes place earlier than day 8? So now we decide to do that. To address that question, now we infect mice, again, black 6 mice, CD40 ligand knockout mice, or CTLA4 transgenic mice. Well, we deplete them of CD4 T cells, then we infect them with flu virus. And this time we harvest the organs at day 5 to look at the total number of antigen-specific CD8 T cells. Just a quick recap of what happened at day 8. At day 8, we have seen that the loss of CD40 ligand or CD28 co-stimulation blocked, both of them has decreased number of PA-specific CD8 T cells. Most exciting thing is, at day 5, as you can see here, the loss of CD40 ligand co-stimulation actually do not affect the generation of PA-specific CD8 T cells as compared to wild type. However, when CD28 is being blocked, the generation of PA-specific CD8 T cells is actually impaired. Therefore, this shows that CD28 actually acts early. It occurs at the earlier time point, but not CD40 ligand. Some of you may have noticed that the scale in the previous slide is pretty low. So the response we had was like 200, 150, 100. So you might argue that, no, this cell number is so little, it's not going to be accurate. So to address that problem, we need a more robust system, and to do that, we use the flu-over OT1 system. So what is the flu-over OT1 system? Basically, we have the genetically modified influenza virus that contains the ovabunin peptide on them. And we have the CD8 T cells that are specific for the ovabunin peptide, and these CD8 T cells are called the OT1 cells. So these OT1 cells recognize the ova peptide on this genetically modified influenza virus, and then they will proliferate. So again, we take black 6 mouse, CD40 ligand knockout mouse, CTLA4 Ig transgenic mouse, deplete them of CD4 T cells. This time around, we infect them with flu ova, which is our genetically modified flu. And 1 day after infection, we put in cell trace labeled OT1 cells. Now, what is cell trace? So cell trace is actually a dye that we stain the OT1 cells with. And as the parent cells divides, the daughter cell now will contain half the die of the parent cell. And with every subsequent division, the daughter cells will always have half the number of the previous parents. So in this way, we are able to track the proliferation profile of these OT1 cells. And now we harvest the organs at day 5 and we measure OT1 cells within the mouse. Like what we've seen with our endogenous PA responses, we observed that the loss of CD40 ligand co-stimulation do not have any effect on the proliferation of OT1 cells. But when CD28 co-stimulation is being blocked, we found that the proliferation of OT1 cells is actually being impaired. And when we look at proliferation profiles, again, we observe that the proliferation is impaired when CD28 co-stimulation is being blocked. So summary 2. What have you noticed at day 5? Firstly is that CD28 co-stimulation affects the early priming of antigen-specific CD8 T-cells. And secondly, CD40 ligand does not affect the early priming of CD8 T-cells. And now to address another question, whether the loss of CD40 ligand or CD8086 actually affects the proliferation of CD8 T-cells. And to do this, we set up in vitro assays. In this case, it's the mixed leukocyte reaction. And what we have here is we isolate CD11C dendritic cell from a box C mouse, and we incubate them with CD8 T cells, either from a black 6 wild type mouse, CD40 ligand knockout mouse, or CD8086 knockout mouse. We then measure the allogeneic proliferation by thymidine incorporation. And as you can see here, if we have CD8 T cells from CD40 ligand knockout mouse or from CD8086 mouse, they are able to proliferate as well as those from the wild type mice. Hence, this shows that the loss of CD40 ligand or CD8086 do not affect the proliferation of CD8 T cells. It has always been believed by many that CD40 exists on dendritic cell, CD40 ligand exists on CD4 T cell. However, our lab recently has shown that during influenza virus infection, CD40 ligand actually gets upregulated on dendritic cell. So now it brings us to the question, is CD40 ligand and CD8086 on dendritic cell affects T cell priming? Again, we do a mixed leukocyte reaction. This time around, we take CD11C dendritic cells from a B6 mouse, CD40 ligand knockout mouse, or CD8086 knockout mouse, incubate them with bulk C T cells this time, and again measure allogeneic proliferation by thymidine incorporation. Excitingly, we found, like what we've noticed for in vivo assays, the loss of CD40 ligand actually results in the decrement of CD proliferation by about 50% as compared to wild type mice. And this reduction is even more dramatic when CD8086 are missing on the dendritic cell. These results therefore shows us that CD40 ligand and CD8086 on dendritic cell are the key regulators in the priming of CD8 T cells. Now, because the past few assays are using thymidine incorporation, the glitch with this assay is that it only takes a snapshot of the cells that have proliferated. We can't tell whether the cells, how much the cells have proliferated, or at which stage the cells are proliferating, or how many cycles of division has gone through. So to address that problem, we label the CD8 T cells this time with cell trace, and we decide to look at the proliferation profiles at days 2 and 3 post-incubation. The reason why we take day 2 and 3 is because our previous in vivo results, as you remember for day 5, suggest that co-stimulation occurs early. So we try to catch the reaction early, so to determine what is happening. And as you can see here, at day 2, when we look at proliferation of CD8 T-cells incubated with CD8 knockout dendritic cell, we found that these CD8 T-cells are not able to proliferate as well as those incubated with wild-type DCs. As you can see here, these CD8 T-cells incubated with wild-type DCs have gone through about 3 cycle division, whereas these guys here are still in the first division. The same trend is also observed at day 3 post-infection. This again shows us that CD8 on dendritic cell is important for the priming of CD8 T cells, and secondly, the effects of CD8 actually acts early in the priming of CD8 T cells. Right. For the past few slides with in vitro assays, we always look at allogeneic proliferation. So what happens during infection? So to do that, now we infect dendritic cells with flu ova, and we put in OT1 cells to look at their proliferation. And as observed here, at 60 hours, the proliferation of OT1 cells is impaired when CD8 is missing on dendritic cells. And the same trend is also observed at 72 hours when CD8 is absent on dendritic cell. And there is minimal effect on CD8 T cell proliferation when CD40 ligand is absent on dendritic cell. So this confirms the results again for the third time that CD8 on dendritic cell is actually important for the priming of CD8 T cells. Right. Previous studies have shown that CD28 promotes CD8 T cell survival. So it upregulates the expression of pro-survival molecules such as BCL-XL. And therefore, we question ourselves whether the decrease of PA-specific CD8 T cell in the absence of CD8 co-stimulation, is it due to apoptosis? To do that, we decide to do a pan staining for activated caspase. And activated caspase is actually a marker for cells that are undergoing apoptosis. And we found that when CD8 is absent on dendritic cell, the percentage of CD8 T cells undergoing apoptosis is higher than the other two groups. For example, the wild type and CD40 ligand knockout DCs. And at 72 hours again, you can see that the proportion of cells dying is actually higher when CD8 is absent on dendritic cell. So the summary so far for in vitro assays. Firstly, CD8 and CD40 ligand on dendritic cells are important for the priming of CD8 T cells. Secondly, CD8 affects the early priming of CD8 T cells, reflecting the results we've seen in vivo. And also the CD8 co-stimulation promotes the survival of CD8 T cells. So the conclusion from this presentation is, firstly, the dependence on CD8 T cell health is actually very much dependent on the stimulant that you use. If you use a non-microbial stimulant, such as OVA, then the presence of CD4 T cells is required for CD8 T cell response. If CD4 T cells are absent, then you're not going to get any CD8 T cell response. However, during some infections, such as those with influenza, if you have CD4 T cells, you're going to get CD8 T cell response expected. However, if CD4 T cells are absent, you are still going to get CD8 T cell responses. And as for viral clearance, the presence or absence of CD4 T cells doesn't matter because you're still going to get viral clearance. So the canonical view that was firstly derived a long time ago is actually pretty misleading because they use non-microbial antigen and they say that co-stimulatory molecules on CD4 T cells are important. Our project has shown that that's not the case. In fact, the direct co-stimulation between dendritic cell and CD8 T cell plays an important role during flu infection. And on top of that, CD28 and CD8 T6 actually acts early in the early phase of primary response, and CD40 and CD40 ligand acts late in the primary response. And so what's the role of CD4 T cells then? CD4 T cells is only required for the development of memory CD8 T cells as shown by Gabriel in 2002. So I would like to thank my supervisors, Andrew, Ifan, and Steve, for their guidance and bearing with my nonsense over the years, being stubborn and stuff, but it was good fun. Emma, Jamie, Robin, Manny, Sophie, and Tim for helping me out during my crazy experiments. Phil for his 8086 knockout mice. Gabriel for sharing her reagents, tips about influenza virus model, her mice, and members of Cancer and Hematology Division for lending me their magnets, their reagents, their tips on apoptosis. Nicole and members of Dorothy Lab for helping out with the virus assays and the tetramer assays. This project started with Laurie in Melbourne Uni in 2002 during my honours years. And when I came back, all the virus assay was actually done by their lab. Ian and Chris from the WHO for growing up my viruses. Of course, I wouldn't be here today without the support from family and friends, and DSO for supporting my PhD studies. Thank you.
Two questions. Firstly, have you tried any other viruses?
Not in my case, but I'm sure that other viruses such as HSV is not going to get this kind of response because they would be CD4 dependent. And if you use LCMV, you're going to get the same as flu. So it's very much dependent on the virus that you use.
And second question is, any idea what the signal is? Is it a TLR, a nod, or something else?
Well, at least for flu, we know that the TLR would stimulate CD40 ligand expression on DCs. So that's one of the methods for flu to bypass CD4 T cell help. That has been shown. For other viruses, I'm sure like LCMV2 can stimulate that, but HSV can't upregulate CD40 ligand on dendritic cell. So we have to further investigate if we want to look at the different viruses and their role in CD4 T cell dependent and independence.
You seem to be able to get it by TLR3 or TLR9, but not the other TLRs. And some, there's one report that says type 1 interferon. There are probably several pathways by which this happens, but as Shirley showed, CD40 ligand is really important.
Yeah.
So for your model, does it require the direct infection of the dendritic cells to bypass the CD4 help, or could you have sort of an adjuvant effect? And if it does require the infection of the dendritic cells, then
Yes.
what kills them? What clears the virus from them?
From ES.
The dendritic cells.
So we know that flu infects dendritic cell. That's one thing, and the direct infection also upregulates like CD40 ligand. There's also things like type 1 interferon, which Andrew has also explained that with strong inflammation, you're going to get CD4 T cell independence. So there's a lot of other pathway other than just direct infection of dendritic cells. Yeah.
Shirley, one of the things CD28 does is will enhance IL-2 production from T-cells.
Yes.
So I wonder in your survival experiment, do you think that's a direct effect of the agent CD28, or whether it's due to the indirect production of IL-2 to promote survival of the T-cells?
I would say it's everything, because I have also looked at IL-2 production in these in vitro assays, and IL-2 is down when CD28 is absent. So there's a few factors other than IL-2 production, which is down, and also without CD28, it also starts to die because, well, previous paper have also shown that 28 upregulates BIM, which causes death. So that's also another factor. And yeah, it also affects the division. So there's a lot of factors that's going on there. Yeah.
Did you try adding IL-2 to counteract?
Yes, I did actually. I did put in some IL-2 in the culture. It did rescue in some sense, but the thing I've noticed is that the proportion of cell increases the proportion of cell that is responding, but the rate at which they divide is still slightly slower than your wild-type mice. So the IL-2 can rescue by increasing the proportion of responding cells, but may not give you 100% at the rate at which they divide to be as efficient as wild-type mice. Yeah.
Are you asking which is the primary reason? That's what you're asking.
Well, this effect on survival could be, I think there is a direct effect of CD28 on survival, but it's hard to tease it out with the
Hmm.
That all interacts like that. If you're asking which is the primary, well, I'm not sure. I know the answer yet.
With your in vivo model, I was just curious as to how effective the antibody that you used was at blocking the interaction.
Of CD28?
Yes.
It's pretty efficient, and it's been used for our transplantation models to show that it actually blocks the interaction. Yeah.
Is that produced endogenously within the cell? How do you produce the
So it's under the rat insulin promoter. So it's produced endogenously and gets secreted and it's in the mouse, circulating in the mouse. Yeah. Questions?
No complaints? Okay, if there are no further questions, let's thank Shirley. Very clear.